And welcome to this IHI Impact Series on partnership joining forces to combat tuberculosis. We have a very exciting webinar today with very distinguished speakers. I will introduce them before giving them the floor. But before that, I'm going to give you a few housekeeping rules. Please use the chat function below of your screen where you can ask questions and interact directly with the speakers at the bottom of the screen. Just as a reminder, this session is being recorded and the recording will be posted on IHI website and on our YouTube channel as well, right before the end of this webinar. So we have started this program in 2019. It's part of the IMI AMR Accelerator. And we are very pleased to show you some results and also to walk you through all the clinical trials that were implemented since 2019. As you know, the drug development process is a very long process. It's a very risky one because you never know whether the molecule you are testing will yield the expected outcomes for the indication you're trying to treat. And also, as you've seen, this program has started in 2019, so the result that you are going to see today that will be presented are the results for the past 6 years, and it's not over yet. So we will hear further on that. Today, we will hear the development specifically of one compound, which is the Alpibacter, formerly known as BVS GSK 098, for those who were at the very beginning of this process. We have started funding this project in May 2019, and then the process has gone to another scheme and came back with us last year. Now, before getting into the clinical trials, I'd like to give the floor to Dr. Fouad Mirzayev, who is the TB treatment and care lead at the Department for HIV Tuberculosis, Hepatitis and Sexually Transmitted Infections at the World Health Organization headquarters in Geneva. He will explain the challenges that we face in fighting against tuberculosis. So without further ado, Fouad, the floor is yours.
Thank you very much, Dirk, for the introduction, and thank you for inviting to be part of this webinar and for this introduction. I see many people on the panel that we know, and it always presenting on the overview of the global situations feels like preaching to converted, but I think it's important to update slightly since the global TB report just came out very recently. First of all, and if we can go to the next slide, TB is preventable and usually curable disease. It's one of those infectious, one of the many infectious diseases, but it's one that is the biggest killer. Cause from the infectious disease. It has left its top place for the couple of years of COVID-19, but since 2023, TB returned to be again. I know, fortunately for that we don't have COVID, but unfortunately TB is the world's leading cause of death again, and it's causing twice as many deaths compared to HIV/AIDS, and more than 10 million people continue to fall ill with TB every year. The incidence decline is not it's steady, but not going down at the rate that we would prefer. It's about slightly less than 2% per year. And one of the important issues, and actually one of the biggest issues on antimicrobial resistance, is the drug-resistant TB that is causing of about 400,000 new cases, back in last year in 2024, and about 150,000 deaths every year. So since I'm going to focus, and we go to the next slide, on the treatment of TB disease, there was quite a lot of developments and the evolution in last 6 decades, when we went from no treatment to treatment with different antibiotics, and the longer just treatment with a few drugs to the multiple drug regimens, switching gradually to the oral regimens and moving towards shorter regimens for both drug susceptible and drug resistant TB, with important increases in the efficacy. There were some drugs that were repurposed for use for TB treatment and prevention. There were some medicines that were discovered for treatment of TB and drug-resistant TB. The direction overall is towards the even shorter, more efficacious regimens, and regimens for all forms of TB, independent of the resistance pattern potentially. And also there are some new developments with the long-acting injectables for TB treatment, similar perhaps of these important and groundbreaking developments with long-acting injectables that we have seen with HIV very recently. Maybe next slide, please. I'll just go through what is not only available but also recommended by WHO, and starting with the treatment of drug-susceptible TB, because most of the patients globally are with drug-susceptible disease, and we have 3 regimens: the 6-month regimen with 4 drugs in initial phase and 2 drugs in continuation phase, the isoniazid, rifampicin, pyrazinamide, and ethambutol, and this is the 6-month regimen that is really used in the majority of the patients worldwide. More recently, we had 2 regimens recommended by WHO. The 4-month regimen that has certain limitations. It cannot be given to anyone below 12 years of age, and this regimen also includes the rifampicin and moxifloxacin, in addition to isoniazid and pyrazinamide. Another 4-month regimen that is also recommended, it's specifically for children and younger adolescents, anyone below 16 years old. It's the same as a 6-month, it's just 4-month regimen with the same medicines. It is probably also important to mention what we have for treatment of some severe forms of extrapulmonary TB on the next slide, like TB meningitis. So that's kind of the area where we don't have a lot of very well based in the evidence regimens. Like, for example, this TB meningitis occurs more frequently in children, so these regimens are more directed towards children. But again, the evidence is not very strong, and it's basically the same 4 drugs regimen as for the treatment of drug-susceptible TB, with just extension of the continuation phase to longer treatment with for 10 months, or the regimen that was recommended more recently, a couple of years ago, based on the operational and observational data from South Africa, that regimen that includes also ethionamide on top of isoniazid rifampicin and pyrazinamide. There is it's important to see some development in this area. The next slide, for drug-resistant TB, we have quite a number of developments more recently, and actually in the last 3 years, with the number of regimens now in the 6-month regimens for drug-resistant TB, which was already groundbreaking, since it brings the treatment of drug-resistant TB to the same duration as the drug-susceptible TB with BPAL-M, the regimen that has bedaquiline, pretomanic, linezolid, and moxifloxacin, and also the second regimen that also can be used in children and adolescents, since the limitation of BPAL-M, mostly because of the pretomanic being a new medicine, not researched in anyone below 14 years of age. There are still quite a number of regimens in the armament for drug-resistant TB, with several, actually 4 different 9-month regimens. 3 of them recently recommended this year, and the longer duration 18-month regimen that is the only one that can be individualized and designed depending on the drug-susceptible, drug-resistant TB pattern. So this is the one as a last resort regimen used for forms of XDR, extensively drug-resistant TB. So now the next slide, let me just show you a couple of the trajectory of what has been happening with the treatment of TB, for drug-susceptible TB and drug-resistant TB. The 4-6 months is actually the duration with which the drug-susceptible TB treatment can be achieved. And while the drug-susceptible TB treatment has been sustainably reached the high success rates for many years, the drug-resistant TB has seen a significant improvement more recently with introduction of the new tools, diagnostics, importantly, new medicines and regimens. So we see a lot of innovation really pays off, because we see this increase from almost 50% global rates of treatment success to 71% as reported in the global TB report released this year. But although the treatment regimens are shorter, but 4-6 months is still very long, both for patients and for the healthcare system. If we go to the next, I think it's important to put that in perspective of the WHO NTB strategy that kind of charted the pathway to the world free of TB, and calls for the 90% reduction in TB deaths and 80% reduction in incidence by 2030, which is in alignment with the Sustainable Development Goals and pushing these targets even further. But this is an important target to go for. But the trajectory on the next slide is what we see is we are really behind that target, because the trajectory to reach that goal should really go to 10% decrease and even further the 17% decrease per year. So, but we have about something in between of 1.5% and 2% per year. So the rate must accelerate, and with the current tools, even with the improvement of and polishing and efficiencies, without new regimens, without new diagnostics, vaccines, and maybe also the long-acting injectables, the shift will not be possible to accelerate this to reach that goal. So what is preventing countries and the world to go for effective treatment? And I can also maybe say a few words about the prevention, because it's also preventive treatment. Um, uh, for TB prevention, there are quite a number of issues with the prioritization of prevention in many places, hesitancy among health workers and patients, and also many concerns about the adverse effects or resistance, since these treatments needs to be given to people who have no symptoms and have no disease at the moment. With the treatment of TB disease, some of the recent advancements, like new regimens as BPAL-M, have showing the promise in improving treatment outcomes. But the implementation remains limited due to access issues, due to regulatory barriers, and policy update delays in many countries. On the next slide, I'm putting also the kind of the diagram that is reflecting what was reported by countries to WHO in the Global TB Report. And this is the implementation of this 6-month new regimen is about 20% of all patients with drug-resistant TB that have been receiving that. But these barriers with the price of the medicines, the patients, not all patients are being diagnosed because the access to diagnostics sometimes is limited. But there are many barriers that are under control by countries and the national TB programs, like the countries still continue use previous regimens. They taking time to update the national policy. They're experiencing some regulatory barriers, and some countries redoing some of the research and the trials that have been done already, if those trials have not had a site in that country. So, but if we go to the next slide, thank you, Tech. We see that now we are at the moment where the pace of the TB treatment and the new regimens and medicines discovery has increased dramatically. And this is kind of a diagram that I put from the recent article by Eric Nürnberger, where it nicely shows what was happening in the new drugs development at the beginning. For the 30 years, there were quite a number of new drugs that came and have been discovered and were commercialized. Then we had like a long drought or the period with almost nothing new, a slight peak, and with the 3 drugs in between 2000 2010, but now we have more than 12 drugs and multiple regimens in the early or later and phase 2 stages of the research. So we are hoping that this will bring some major innovation into the field of TB treatment and hope maybe also TB prevention. And if we go to the last slide, we are in WHO very much looking forward to see this research into these new chemical entities to move to the regulatory approval, and we'll be happy to do what is needed to work on the public health policies, and not only make sure that the drugs and the new regimens and treatments are available, but they also reach patients in those countries that are need that the most, and patients that need the most. So with this, I want to wrap up and thank you all for the attention, and happy to stay on and answer questions if needed.
Thank you very much, Fred, for this excellent setting the scene of TB, presenting the challenges that TB is still today in our country is very important, and that helps us. So now we will give you more insight about clinical trials and let you know everything you wanted to know about clinical trials without asking. And giving the floor to Dr. Michel Pirenne, who is the clinical program team leader at BioVersus. And he will tell us the development of their vision to propose a new molecule and how they managed to determine the mechanism of action. Michel, the floor is yours.
Can you hear me?
Yes, we can hear you.
Okay, thank you very much. Yeah, next slide, please, Tech. Also, this one we can, yes, thanks a lot. So what we will cover in the next 3 presentations is basically the timeline from 2019 to the end of this year. I will start off with the preclinical work, including the phase 1 of this Alpibectier molecule. The program was called TRIC-TB. We will then hear from Andreas about the phase 2A trial, which was funded by EDCTP. And as Tech mentioned, it came back to IHI with a program then where we called them Enable and Step 2C. It is a very successful public-private partnership. And on the bottom of this slide, you see the rough cost sharing. Of course, we got a lot of support from EU-funded agencies, IMI, IHI, EDCTP. Nevertheless, if you want to do successful drug development, it needs also 2 private partners, which will invest a lot into the program. And we have a rough cost estimate from BioVersus, GSK, and EU partners of roughly 1/3. And I think this really makes what brings the program forward. On the next slide, we start off in explaining a bit how this whole project actually started. And you see it on the top left, already in 2012. A university group in Lille and the Institut Pasteur in Lille were working on a new mode of action, a new molecule to tackle actually antibiotic resistance in TB. At the very much same time, also BioVersus was engaged in this novel transcription regulator. And in 2012, we then partnered with the group of Alain Bollard, Nicolas Villon, and Benoît Després at the academic side, trying to bring this program forward. And as early as in 2014, then GSK entered into this play as well. I would say this is quite early for a big pharma company to go into a deal at the hit identification stage. But since then, let's say the 4 of us moved this program forward. And then in 2017, the number of molecules that eventually make it into the clinic became less and less, and we have selected 2 molecules out of the battery of molecules that we have tested, which we wanted to profile a bit better. And with GSK entering into that collaboration, Wellcome Trust money entered as well. So you see it was a public-private partnership already at the very beginning. And now this is then in 2019 where we started off with TRIC-TB and we got this funding from IMI. It was BioVersus and GSK, only 2 partners in a big EU-funded project. We kept our academic collaborators from the Institut Pasteur Lille and the University of Lille very close to us. That's maybe one of the take-home messages. Never leave the brains also behind the research. Try to keep them inside the consortium. It makes your research in general stronger. So we started then off with the identification and profiling of this molecule, which is called Alpibectier. And on the next slide, I'm going to dig into a bit the mode of action of how this actually works. So what we try to do is to boost the activity of a very old antibiotic, ethionamide. You have heard the name maybe before from Fuad. Ethionamide is used since decades for the treatment of drug-resistant TB, and it's also used for the treatment of TB meningitis, but it has some liabilities, as we will see later. And the discovery of this new molecule, Alpibectier, was really key to rejuvenate ethionamide, because it brings some very important features for making this drug successful. And it will always be a combination. We need ethionamide as the drug that kills the bacteria, and we need Alpibacter, which makes ethionamide great again, if I can say it in this word. So only the two of them, Alpibacter and ethionamide, are finally active against TB, and we try to position it for pulmonary TB, so if you have infected lungs, but also for TB meningitis. And on the next slide, we must first speak a bit about the old drug, which is ethionamide. I mentioned it is used since decades for the treatment of TB. Now not really in favor for the clinicians anymore. We have new and better drugs. Maybe the most drawback from a clinical point of view is the gastrointestinal intolerances. You can use doses up to 1,000 milligrams, not grams as stated here, 1,000 milligrams daily, but this dose is often poorly tolerated, and you have to imagine TB patients not only take one drug, they take several pills every day over months. So you get side effects, and on the right-hand side, you see a bit how these side effects actually influence the treatment discontinuation. If you are about to take 1,000 milligram daily, over months, you have a lot of patients who will not successfully complete the trial. So ethionamide would be a good drug, but currently the GI intolerances is limiting its use. And to understand what ethionamide does, we need to look into the bacterial cell a bit closer. So what you see here in the circle is M. tuberculosis. That's the bacteria that actually causes TB. You see ethionamide pointed on the left side. Ethionamide itself is not directly killing the cell. It is bioactivated by an enzyme. It's the Pac-Man-like structure called ETHA. This enzyme converts it into a preform that then couples with NAD+ and forms an adduct that finally inhibits the target INHA. And if this INHA is inhibited, the bacterial cell dies. Unfortunately, this bioactivation of ethionamide is very insufficient. There is a transcription regulator, you see that ETHR, that basically sits in front of the expression for ETHA. So there is only little Pac-Mans available, only a few Pac-Mans that convert the drug into an active form. So this means consequently that you need a lot of ethionamide, so a high dose of ethionamide, if you want to kill your bacteria, and this causes the dose-related side effects. Another drawback is that after maybe 60 years of use of ethionamide, you have some resistances in this pathway, ETHR, ETHA. So even if you would have expression of the enzyme is mutated and cannot make the antibiotic active. And this is where Alpibacter now comes into the game on the next slide. So we have discovered novel bioactivation pathway. Ethionamide can not only be activated by ETHA, but also by MIM-A, for example. That's now the green Pac-Man. Also, MIM-A transforms ethionamide into an active drug. And Alpibacter binds to a transcription factor which is called VRS. And by doing so, it stimulates the expression of MIM-A. So now we have a lot of Pac-Mans inside the cell. Ethionamide is very easily transformed into the active drug. And with this, we can lower the dose. We can now reduce the dose of ethionamide and still be efficient. So we get rid, hopefully, of the gastrointestinal side effects. And because it's a completely alternative pathway, we are also active on resistant strains. And this, I think, is shown on the next slide. So now we look a bit in microbiology. Here we have tested 100 different clinical isolates of Mycobacterium tuberculosis bacteria. And we asked the question, what is the concentration of ethionamide that we need to inhibit the growth of 90% of these strains? And in gray, you see the curve for ethionamide alone. So here you would need more than 8 milligram per liter of ethionamide to have a 90% coverage. Now, in the presence of alpibectir, we boost the activity of ethionamide. So you see that clearly the curve is shifted towards the left, so towards lower concentrations of ethionamide. And now already at 1 milligram per liter, we kill or we inhibit the growth of 90% of the bacteria. And lower concentration hopefully translate into a lower dose as well. And we are not only active on drug-susceptible strains, we are equally active on isoniazide-resistant strains, multi-drug-resistant strains, ethionamide-resistant strains. We are also active on bedaquiline and pretomamide-resistant strains. So very potent antibacterial activity. And now this is the proof of concept in mice. I only explained the left-hand side. Here we have infected a mouse with TB bacteria. You see the number of bacteria on the lung on the very left with this 5.4, that's a logarithmic scale. So quite some bacterial burden on the lung. If you do not treat these mice with anything, you end up with the dark gray bar, so a very high bacterial burden on the lung. And then we used isoniazide, which is kind of the gold standard antibiotic to see bactericidal activity. And in the presence of isoniazide, we can reduce the bacterial burden by 1 log. And ethionamide alone at 50 milligrams is almost as active as isoniazide, but not exactly. And then you have the 4 blue bars, and the one which has the 0 at the bottom is 15 milligram per kilogram ethionamide. So this is now a third or even less of the dose of 50 milligram. And you see, of course, alone it is not very active, but then if we give alpibectir on top, you see that a dose response curve happens, and we can significantly improve the killing of this bacteria, and we end up with a combination which is equally active or even better as isoniazide in this mouse model at 1/3 of the ethionamide dose. And then on the next slide, that was for us a highlight to see that alpibectir not only goes into the plasma, you see here a plasma profile in rats, you see the concentrations over 24 hours, but you also find the molecule in the CSF, the cerebrospinal fluid, and in the brain of these rats, so clearly indicating alpibectir penetrates the blood-brain barrier, and that could be very important if we want to treat TB meningitis. And with this information, finally, we then started a clinical trial that was now funded by TRIC-TB. And in infectious diseases, a clinical trial phase 1 is normally in healthy volunteers. You start very low with the dose, and then you successfully increase the dose. You see we had 0.5 to up to 40 milligrams. Most of the participants, they get alpibectir, but you also need to have a placebo control. And then we escalated the dose. First, we only gave 1 dose per day. And at the end, we even gave 7 doses on 7 days. So one dose daily over 7 days. And you see the capsules that we have produced on the right-hand side. This is truly the alpibectir product in the capsule that then the healthy volunteers swallowed. And we have assessed all the safety parameters. And on the next slide, you see from a safety point of view, it's a very high level bullet point summary, but we did not have any SAE. None of the participants had to stop the treatment. No subject has withdrawn from the trials. There were, of course, some AEs happening, but it was not dose related. These were the headaches, for example. Nothing, most of them were mild or moderate, and overall a very good clinical safety profile. And on the right-hand side, you see a bit the pharmacokinetics of these participants. You see the drug peaks after every day that you give the drug, and then it falls down, and then on the next day they took another pill, so the level is increasing. And in red, you see what we have predicted from the mathematical model. And in black, you actually see the exact concentrations of the participants, and you see the model very well predicted the PK of this drug. And with this, we were very happy to see the phase 1 successfully terminated. You will now hear about the other programs. I just want to conclude with this message. We got until here. You see the sailing boat. We are crossing the ocean, and we are committed to also do a phase 3 trial. And let's hope that also the public side of this partnership doesn't end here, and that we are not stopped without wind in the middle of the ocean, but hopefully can continue this message until the very end, until the approval for that drug.
Indeed. Thank you very much, Michel. And as Michel said, this is a long process, and with this very promising result from the phase 1 clinical trial, then that moved to the presentation of phase 2A clinical trial by Professor Andreas Dijken, who is the founder and chief scientific officer of TASC Clinical Trials in South Africa. And the beauty of this presentation is really to show the complementarity between what IHS has done, because part of the presentation that he will do now is also funded by the European and Developing Countries Clinical Trials Partnership, EDCTP2. So, Andreas, the floor is yours.
Thank you. I've been told that I can't move the slides, so somebody will have to do it. Thanks. So this is it, and I actually quite like that sailboat because, you know, down the Rhine from Basel and then into the sea and down to Cape Town, it takes a while, but it did arrive eventually at my trial site, this drug, and I was quite happy about it because I was with the thinking about it for many years before when the results were presented about it at conferences. And I thought that this is a really fascinating concept. And I sincerely hoped that I would ever get my hands on it to do a clinical trial with it. So that's what we did in Cape Town. I can show the next slide. There is obviously some overlap with what Michel told us. So you've seen this slide before. So you put two drugs together and you get more than you get from each added, right? So I think the two little ones here give a much bigger pill than you would arithmetically expect. I don't really understand the chemical formulas up there, but I can tell you these are not complicated molecules to make, which has a good bearing on the cost that they might at some point have if they work. So what Michel told you is that isoniazid, as he called it, you can go on back, or you can leave it here, it's fine. Isoniazid, as I will always call it INH, we clinicians call it INH, is sort of the benchmark for this class of drugs because ethionamide and isoniazid share a common target. And the target is, I believe, it has a top level that you can do with it. And in isoniazid, we know that giving more drug to people eventually doesn't do more. So at somewhere at 300 milligrams of isoniazid, we reach a ceiling. And so for us, for a human experiment, we would be happy if we can reach that with either ethionamide alone or boosted ethionamide at a nice combination. So that's to start out of how we think what we want to see in humans reproduced. And you see on the right side, this is what happens when you give humans ethionamide. They don't like it. And this is a big difference between humans and mice, is that mice don't complain. Humans do. And if you're not there to convince them every day to take it, then they just won't. And in tuberculosis, you can't hospitalize everybody to supervise treatment compliance. So you have to give them a regimen that they eventually take. And if there is a drug in there that they don't like, they can't necessarily see, there, is it the green ones or the gray ones or the long white ones or the little white one there? Which one is it that makes me feel so bad? And then they will stop taking all the drugs altogether. And this is what we really want to avoid. And this is why this is such a big step forward if this works. Okay, next slide, please. Right, so this is the mouse model. Now, Michelle explained to you very nicely, okay, if we don't do something which we can't do in humans, right? You can't do a placebo trial in a tuberculosis patient. That would be unethical. So the only benchmark we can really use is that isoniazid black bar there. We need to do something that tells us we can reach this. And the other things we can probably do, we can give people ethionamide at a maximum dose, at a lower dose, and we can play with the doses, a lower ethionamide, a bit less Alpibacter, and then more Alpibacter, less ethionamide, and all that. So it's not too difficult to figure out how to do this, to find it out, yet funding is limited. So one can't do everything. So if I summarize the text quickly, we do know that ethionamide activity is dose-related. More ethionamide will give you both more activity, but also more gastrointestinal intolerance. Isoniazid activity is dose-related and it has a ceiling. And the maximum activity of both is similar, but ethionamide is not well tolerated. So that's what we knew. And what we needed to find out is if Alpibacter can lower the ethionamide dose, and what the right combination would be between Alpibacter and ethionamide so we can hit the INH level without the side effects. Okay, next slide. So many, this is now the nice slide. I'm showing you some pictures from South Africa. So just to set the scene, I'm not entirely sure where you're sitting, but you're probably not in a TB endemic area. So this is a waiting room in a TB clinic, in one of the 100 and something TB clinics we have in Cape Town. So there is a lot of that disease here. And this is the reason why it's a good area to do clinical trials in tuberculosis patients. About half of our patients are also HIV co-infected, so we can also test that interaction. Next slide. So what we do have here in Cape Town is this wonderful institution that we can only keep running because these studies keep coming and we can use the funding to keep the lights on, so to speak. What we have in South Africa, we have a lot of doctors like the one you see there who really know their way around tuberculosis. They know when tuberculosis gets better and when it gets worse. They have this kind of clinical feeling for it, much more than any doctor in Europe can ever develop because they just don't see the disease that frequently. Next slide. And so in this unit that we have built over the years, we have 24 beds entirely for TB patients that take novel drugs under close supervision by our staff. So they basically never leave the unit because you don't really know when it's a new drug if it works at all. So anything can happen anytime, and we need to be able to evacuate these patients to a university hospital or the appropriate place if something goes wrong, which happily never really, not very rarely happens, let's say. So we have become quite good at it. So this place is ventilated a bit like an operating theater. You have 6 to 12 air changes an hour. So in here, it's probably safer than outside when it comes to tuberculosis because we're cleaning the air so frequently that you could even do surgery in these rooms. Next, we separate the area clearly from the in and outside. So these doors where you see this lady coming out here, these are 2 doors that never simultaneously open. So you can go into that intermediate space, the one door closes and then the other door opens and you can go in. So there is never an air transfer between the staff area and the patient area. And if you have to go in there, you wear an N95 respirator mask. Next slide. we have fortunately also have all the support that you need. We have laboratories, we can do drug levels in blood, we can collect all these samples, freeze them at -40, -80. It's an incredible infrastructure that needs to be maintained for this kind of work. One can never forget you can only do this once. A clinical trial you cannot repeat. Every mistake you make, it cannot really be corrected, so it has to work. Next slide. We have a pharmacy to look after these drugs. These are non-registered drugs that have specific legislation. They're basically illegal substances, and one has to have all the permits in place. And there will be frequent inspections about what happens to these substances. And I have 4 or 5 pharmacists there that do this for a living, and they're very good. They basically also being used close by academic institutions to keep their trial drugs safe. Next slide. But what we can also do is we have our own kitchen, so we can make sure that the predicted amount of calories and fat contents in the meals is similar to what the healthy volunteers in a previous study had, so that we are not interfering too much with the absorption of the drugs. There's very little experience with this, when they bring the drug to our site. So we follow the instructions of the protocols very, very strictly and very clearly to get the results that we want. Okay, next. Can you move the slide? Thank you. Then there is obviously all the back office of it, with data capturing and meeting rooms and monitoring and audits and all that kind of thing. So we can go one further. What do we actually do? So we find people that we call the healthy TB patient. So that sounds funny, but you basically, to do a clinical trial safely, you need to find a person that has tuberculosis but isn't very ill from it, because that's a big risk. If you have a really ill person and then you start giving them unregistered medication, it's risky. But at the same time, we need a person that has very severe tuberculosis, like the one on the chest radiograph there, because we need a lot of bacteria that they cough out so that we can measure how well the drugs reduce that count over time. So we basically need to find really people with really bad radiographs that are not very ill. And that's like a needle in a haystack. And for, let's say, 50 patients that we look at, we can only use one. But we have so many of them that we successfully can actually do this kind of study. And the point is that we collect sputum at the start for 2 nights, and then we start giving drug and we keep collecting sputum, and we keep measuring every day how many bacteria we can find in these sputum samples at the middle picture on the right. And when a drug works, they will drop, the counts will drop, and when the drug doesn't work, the counts will not drop. So that's the proof of concept. And the thing is that we don't really know until 6 weeks later, when the cultures have grown, what is actually happening. And so this proof of concept for BioVersus, you can move on. Sorry for confusing you, Tech. Here we went about it. I mean, it took us probably about 20 teleconferences to finally agree on how to do this, because obviously you would like to do about 20 groups to find the exactly right mixture of ethionomide and Alpibacter, but you just can't do that because there isn't enough time and there isn't enough funds. So we started on the left top, and we gave isoniazid to be sure our lab assays work more or less, and we know what to expect with isoniazid. And we gave a quarter dose, third dose ethionomide with a little Alpibacter to very carefully start the study and see what happens. And then we collected all the data, went to the independent committee and made a decision, right, this isn't dangerous, and we can move on. And then we went to the part B over there, where we completed the controls with isoniazid, and we had a That's at the bottom, the gray boxes. We had a low dose of ethionomide and a high dose of ethionomide. So if you wanted to not be randomized into one group, it was the bottom one. That's where you got the full dose of ethionomide that we would predict that you had severe side effects. And of course, the patients knew this, and we couldn't influence who went where. And usually the patients are quite happy to participate in these experiments because they know it's for a good cause, and they report for a week and then it will all be better. So we went through all this, isoniazid as the benchmark, low dose ethionomide that shouldn't do too much, but should do something, high dose ethionomide that should be more or less on par with isoniazid, and then 3 combinations of Alpibecter, now a higher dose this time with a low, medium, and higher dose of ethionomide. And we did all this, and what we expected, if you go to the next slide, was this. So this is basically what we wanted. And here you need to know that the drop is important. So from the 0 line at the top, a drop of 1.5 in a week with isoniazid would be okay. You know, that would be the benchmark. With ethionomide, the second dot there, the green dot, it should drop a bit, but, you know, by far not as much as ethionomide would. With the full dose of ethionomide, for those patients, poor patients that got into that group, you would expect a similar effect to the isoniazid one. And then with the mixtures, so with Alpibecter at the lowest dose and 250, you would expect more than with the green 250 on the picture, because Alpibecter is now supposed to improve it. And then the second one from the right, orange, with the higher dose of Alpibecter and 250, you should almost reach isoniazid. You should certainly reach isoniazid with the one on the right, where the high dose is given with a high dose of ethionomide. And if you give a high dose of Alpibecter, that's the third one from the right, with a very low dose of ethionomide, you should see still a better effect than 250 of ethionomide, but perhaps not the full effect. That would be very cool if that worked. But from the biological theory, it would almost be impossible. So the next slide should be the side effects. So this is what we would expect. I mean, obviously we did capture this in much more granular fashion. There's every day the doctor goes through to every patient to ask them very specifically how they feel. And there was a specific questionnaire for gastrointestinal side effects that was validated in many languages and that we consequently used in every patient. But here, very roughly, with isoniazid, we would expect few things to happen, and with ethionomide 250, just as few, even though that's a low dose, right? Then with 750, we would expect to see side effects, complaints frequently. And then with other mixtures, it would vary on the amount, basing on the amount of isoniazid. But the sweet spot would be where you get the highest activity with the lowest frequency of adverse events. Next slide. So this is scenarios that we thought up front that we would think were not good. So the top one, if Alpibecter did not boost ethionomide at all, this would result. You see, no orange ball gets anywhere close to where it should be. At the lower one, if ethionomide, which was actually a difficult drug to source, it's so little is made and consumed on this planet nowadays that we struggled to actually find some and buy some from the Green Light Committee and from other pharmacies that were still producing it. So we weren't actually sure if the ethionomide was super quality. So if it wasn't, that's what you would see, you know. So I'm not sure if somebody told you before, but we did this experiment. The study went well, but we are currently not allowed to show you the results because they are submitted for publication at the journal that is very specific about not disclosing results to the public before it's been published. And we don't want to jeopardize that, but because there is actually another speaker and because we have been invited to give you this talk, you might assume that the experiments weren't one of those outcomes, but that's where I have to stop telling you how it went. But I went through the great lengths to try and explain the thinking to you. And the next one, that would be how the experiment could fail as well. So if with even the lowest doses of ethionomide and Alpibacter, you get a lot of adverse events, which would sort of mean that Alpibacter itself, even though it's given at a very low dose, somehow makes these people feel bad. You know, that would also make us, disappoint us. Okay, next. I think there's only one summary slide left. That is the next project that we had planned we would do if this one was successful. Simon will tell you a lot more about it. I'm just going to jump one over and come to my conclusion slide. So this was the BETO study for proof of concept, which should have given us the confirmation that the mice don't lie. You know, even though they can't complain, at least they didn't lie to us, and the biological principle worked. And the next study that Simon will talk about will then tell us more what this can do in a combination with other drugs, because you can never treat tuberculosis with only one drug. And the very next thing then is that ALP-E, as I now like to call it, which to me is a new entity in its own right. You know, it's a fixed combination of that is like a new drug. It should no longer be called ethionomide. It's now ALP-E. It's a new agent. It could replace isoniazid in standard regimens, and it could be used for TB meningitis and perhaps other regimens in drug-resistant tuberculosis. And that's where I stop and hand over to Simon.
Thank you very much, Andreas. And actually, you put the seed for a part 2 of your presentation once the publication is out, so that this is the great trailer for the next step. So now I'm going to give the floor to Simon Tiberi, who is the Senior Clinical Development Director at GSK, and he will tell us the next steps for the development of this ALP-E. Thank you very much, Simon.
Thank you very much for the kind introduction. Thank you for the invitation and also giving us the opportunity to really bring TB to IHI audiences. Next slide, please. So I'll be following up from Andreas's story. So the BETO study, then progressed now to enable, and we're back into IHI now from EDCTP. We've moved back to IHI to a very large IHI funded project called Unite 40B, which is a phase 2 platform trial organization. And it's funded thanks to IHI, also from FPIA partners and also from DZF. And next slide, please. It's a very large family making up the Unite 40B consortium. So it has a very high level of expertise and experience in the TB space. And so it really takes a village, really, to get out there, get new TB drugs, and hopefully really accelerate the development of TB drugs. And from the previous discussions, generally it takes decades to get these drugs to patients. Next slide. And so the idea of Unite 40B is really not only to get these drugs individually out there, but it's also, in the meantime, while we are evaluating these drugs, is evaluating these drugs together. And so the hope is that through accelerating, thanks to this very innovative platform, we're not just evaluating one drug. Here we're evaluating, actually, this is an old slide, but we're evaluating 6 new chemical entities within the Unite 40B platform. And the idea is that we're not only developing individually single drugs, but we are also evaluating these single new drugs together to really formulate new regimens that will really change the trajectory for people living with TB. So we want to have safer, simpler, faster regimens that will really make a paradigm shift in the way we treat TB and really help the WHO in eliminating TB more quickly. So, the idea is that these drugs are selective. They're not repurposed old drugs, but they're new drugs. And Alpibacter is refurbishing an old drug like ethionomide, it's a really smart concept. So from here, next slide, please. We have the ENABLE study which in 2025, so it happened in 2025. I'm happy to say, thanks to Andreas. Andreas's site was the sponsor of the ENABLE study, and it was conducted at 2 trial sites in Cape Town, UCT and TASC. And the last visit was on the 11th of September 2025, so it's happened as well. Next slide, please. And from the ENABLE study, we also have now our sights on going to a phase 2b study, which is called STEP2C. That's also funded via the EDCTP through the Panacea network, but also now via IHI and the Unite 40B network as well. Next slide, please. So these were the current treatment timelines. They've changed slightly, but what's happened is ENABLE has taken place this year, and it has completed, and we're currently working on getting the results and seeing those results. At the same time, STEP2C, the timeline at the bottom is we expect to start recruiting and treating people living with TB in Q1 of 2026. Next slide, please. So within Unite 40B, we have 12 work packages. And from these 12 work packages, which divide up all the work within the Unite 40B consortium, we have a work package called Work Package 4, which is led by Professor Diacon. And its mandate is to conduct phase 2A clinical trials, which is looking at early bactericidal activity of the different drugs. And 3 to 5 new chemical entities will be going through the EBA platform that's at TASC and also at UCT. And the idea is that, you know, we really define the proof of concept for these drugs. Does it work? Does it kill TB? And also get the safety package, but also understand the dosing as well. So what's important here is that we also are able to determine what is the most effective dose that kills TB, and also what is the best tolerated dose for TB. And with this in mind, this is where ENABLE really shone through in the work package for. Next slide, please. So this is the ENABLE study. This is ethionomide apobectir for pulmonary tuberculosis. The sponsor's TASC, the sites at TASC and UCT in Cape Town. It's a phase 2 randomized open label study, 2 sequential cohorts. The idea for the sequential nature was really to ensure the safety of subjects, because obviously these are new drugs. Through the results of BETO, we had a suggestion potentially that we hadn't reached the highest dose of apobectir, and potentially we could get more bactericidal activity by increasing apobectir further. We didn't know, and so the idea for ENABLE was really to create a dosing curve, not only for ethionomide, which was conducted with the BETO study. So thanks to the BETO study design, we're able to have a dosing curve for ethionomide, but we didn't have a third dose for apobectir. So we had 9 milligrams and 27 milligrams with the BETO study, but with ENABLE, we were including a third apobectir dose, one of 45 milligrams. And so ENABLE really complements the understanding from BETO, allowing us to, one, increase the dose of apobectir further. And the idea here is that, you know, potentially increasing the dose of apobectir could potentially allow us to drop down the dose of ethionomide further. And so, you know, that's potential hope is that, you know, instead of reducing it threefold, we could potentially reduce it sixfold or more. So that was the hope or the idea between the design. And then once we've recruited cohort 1 with apobectir 45, 125 or 250 of ethionomide, and obviously with a control arm being HRZE, so the 4 drugs. This is a difference with BETO study, where we only used isoniazid as the comparison. This study also allowed us a 2-week study. So BETO was only a 7-day EBA, Enable is a 2-week EBA. So we're getting also safety information here. And cohort 1 was really just ensuring that we were safe at 45 milligrams, and we were safe to then include 45 milligrams of Apobector, 250 milligrams of ethionamide, and then combine them with rifampicin, pyrazinamide, and ethambutol, with the idea that, you know, potentially we could replace isoniazid and finally treat isoniazid resistant TB, which is currently a massive gap and an unmet need in many countries around the world with high frequency, high prevalence of isoniazid resistant TB. Next slide, please. So the objectives here were to evaluate the EBA, the early bactericidal activity, the safety, the tolerability, and the dose response. Establish the safety of 45 milligrams of Apobector with a cohort 1, and then having an interim analysis there to see whether that was safe or not, and just from the safety perspective, and then also a PK/PD analysis of the Apobector ethionamide dose titration. So taking already what we've already did in BETO, and then complementing it with the 45 milligram dose of Apobector. And then also to establish the safety of combining Apobector ethionamide to the rifampicin, pyrazinamide, ethambutol combination. So there have been some concerns in the past of including ethionamide in combination with rifampicin, pyrazinamide. There was potentially the main concern was combining rifampicin with ethionamide, which was seen in leprosy studies in the 80s where there was increased liver toxicity. So because of that, label, we decided that we had to generate safety information and safety data. The evaluations were time to positivity and colony forming units, so liquid culture and solid culture for Mycobacterium tuberculosis, the Mycobacterium assay at the MBLA, and the PathFast LAM tests, the 22 gene transcriptomic signature conducted by Borstor, and we were evaluating safety, tolerability, and pharmacokinetics. Next slide, please. So the sample size was 15 smear positive TB patients per arm, so in the 45 125, 45 250, and then 15 further PTB patients in the RZE Apobector ethionamide arm, and we had a control arm with HRZE, 15 patients there as well. The duration was a screening of less than 9 days, the 14-day EBA study with the 2 stages, and the 2-week follow-up. And we were evaluating 14 days of activity. We thought that was more suitable than the 7-day one, so that would give us more safety information. And then our GSK safety board endorsed the 14-day EBA study. And then we have a flow diagram below to visualize it. Next slide, please. So the objective was really to see whether or not we had early bactericidal activity, and this is combining the data with BETO. So the big question was, does increasing the dose of Apobector give you even more bactericidal activity, yes or no? And also, is it safe? And this was 2 centers, and we extended to 14 days, and we had 60 participants. Next slide, please. So that was the primary endpoint, TTP, so time to positivity over 14 days, 0 to 14 days, so we can see a slope. And the idea is that if it's very bactericidal, the slope will be more acute, and if it's less bactericidal, then it's flatter. Secondary endpoints are also looking at the colony forming unit, so the solid culture, and we're also evaluating the early bactericidal activity 0 to 2 days and the early bactericidal activity 2 to 14 days, evaluating safety and tolerability and looking at the PK analysis. And exploratory endpoints, we're looking at biomarkers, mycobacterial parameters, drug-drug interaction, and PK/PD analysis. Next slide, please. So inclusion criteria were written informed consent. We included both male or females between the age of 18 to 65 years, weight between 40 to 90 kilos. They were all newly diagnosed and untreated for pulmonary TB. They had to have susceptibility to rifampicin or isoniazid, and that was evaluated on the expert and on the expert XDR. Chest X-ray consistent with TB, and an expert positive with a quantitative readout of medium or high. And we had to have people with TB who could produce sufficient amounts of sputum for the study. We also required the female participants to be of non-childbearing potential, or if they were of childbearing potential, to use effective methods of birth control. Next slide, please. The exclusion criteria is a very long list, but many of them were around whether or not they had QTc issues in the past, whether they were too sick to enter a clinical trial. Those were the main ones. Next slide, please. Biomarkers, we looked at the MBLA at baseline at day 14 and the PathFAST-LAM at baseline day 4, day 8, day 15, and the 22 gene transcriptome signature model. The reason for including biomarkers is because there's a real gap in diagnostics, and we need to have also better endpoints in clinical trials to give us a better idea of, a predictive model of whether these drugs really work or not, and what essentially can they deliver. What can they bring to the clinic that other drugs don't bring? And so we do need to really evaluate new biomarkers. So the hope is that we also have in the future new biomarkers, also thanks to studies like Enable and Unite 4TB's Paradigm 4TB. Next slide, please. We also have PK sampling in the study, and we did intensive full PK at day 1 and day 14. And trough sampling on day 7, 9, 11, and 13. Next slide, please. So currently the study progress, we had the last subject last visit on the 11th of September. There is data that's being transferred currently from TASC to GSK, where we will be ready to evaluate the microbiological data and the PK data. And so we hope to then be able to get a dose for Alpobacter and ethionamide for the STEP 2C study, which we hope will start in Q1 of 2026, which is a phase 2B study. Next slide, please. So in summary, we believe the BETO established proof of concept for Alpobacter ethionamide. Enable will refine the Alpobacter ethionamide dosing. And what is next for Alpobacter ethionamide? We hope that we'll be able to start with the STEP 2C study soon in Q1 of next year for pulmonary TB, and BioVersus should also hopefully start their TB meningitis study shortly as well. And thank you for your attention. I'm happy to take any questions. Tech, you're muted.
Thank you very much, Simon. I had issue with my mouse actually, so I can't. I'm supposed to stop sharing, but I can't now. Thank you very much, Simon. And we have the time to share a few questions from the audience. I'm going to log into the session. And so we have a first question now. As Fouad is still with us, it's a question that could be answered by Fouad and also by Andreas. What are the main reasons the global TB response remains off track, as Fouad has presented earlier, despite the availability of improved treatments and diagnostics? So do you have any lessons to share? So you alerted a bit about that earlier in the presentation.
Yeah, no, maybe from my side, just to reiterate what I tried to show on the slides, that with the available tools, with the treatments being really too long, with diagnostics that are not point of care, for TB, we can do as much as it is happening now. So some change in the curve to get to the elimination, which doesn't mean no TB at all, but it's very low levels of TB incidence globally. We need something revolutionary, either really the new treatment that is very safe and very short and very effective, coupled with the point of care diagnostic that has a very high sensitivity and very specific, or the vaccine that works for everyone, even post-exposure, or any combination of these new very revolutionary tools to get there. So that's the main reason.
Thank you, Fouad. Anything to add from the panel? Or we move to the next question? Thank you very much, Fouad. So the next question that we have is, what are the key ethical and operational challenges in conducting phase 2A early bactericidal activity studies in high burden settings? And how did your sites in South Africa help to overcome them, actually?
Yeah, I guess that's a question for me. So, I mean, I'm stating the obvious upfront. All our trials are approved by our regulatory body in the country, the South African Health Products Authority, and also by at least one independent ethics committee. And these we are frequently inspected to make sure we actually do adhere to these regulations. Then there is the more philosophical aspect of it. And I've learned quite a few things since we do have a community advisory board, which is more than 10 years ago. There was an initiative from the Global TB Alliance to go out there and find tuberculosis patients or people that are directly affected by the epidemic and actually ask them about their feelings about the ethics of this. And it is actually quite surprising that they feel that we are not, let's say, progressive or aggressive enough in our approach as researchers. They feel that we are applying first world standards to the drugs that should be used in a third world environment. So we are looking at a minute risk of a cardiac side effect that you can only see on the ECG, and no one knows if it has any real clinical consequences, whereby we have 1 million people on the planet that die despite getting the best available treatment. And that we should perhaps consult the patient organizations more frequently, and they would probably be encouraging us to be a little more, how can one say that? To be a little more forward when we start bringing novelties to them. So all that said, I think at least in the South African environment, where the minority protection, you can imagine after previous political situations we had here, is very strong, that the sentiment in the clients that we actually have is that we are almost overdoing it a bit on the ethics side, that we're doing too much to risk nothing and slowing down the process, and we're not bringing novelties to them fast enough. Okay, that's just the ethical aspects. I mean, Fouad will have to tell you a lot about the lack of funding and political resistance and social issues and all other things that need to be fixed. But looking at the ethics, I am very confident that we are here not exploiting any minorities, and we're not really, doing things that I could not 100% stand behind. But you will have to believe that. I can only give you my own opinion.
Thank you very much, Andreas. Another question that we have received is, how might Alpibacteria's novel mechanism of action change future drug-resistant tuberculosis treatment strategies especially in settings with high levels of ethionamide resistance or toxicity concerns?
Maybe I can quickly, at least from the ethionamide resistance point of view, that was shortly shown on one slide. So we checked a lot of strains which are resistant to ethionamide, but as mentioned previously, the ETHA pathway is the predominant pathway to bioactivate ethionamide, and we fully overcome the resistances to ethionamide by using this alternative pathway via Alpibacter. So we are very confident that the drug works on ethionamide resistant strains as it does on isoniazid resistant strains too. So I think there is a place for this drug as well. You know, it's a rapidly cidal drug combination, and I think there is a place for such a drug also in a drug resistant regimen.
Thank you very much, Michel. There is a question from the audience, and I think that this one may be for Simon. Is Unite for TB also open to looking at vaccines projects?
Thank you very much. From my understanding, it's a drug platform trial at the moment, but it does have a lot of sites, and it does have a lot of expertise and work packages. So I would never say never, but currently its real objective is really to bring new drugs and new drug regimens to people with TB as fast as possible. So it wouldn't really fit well with the kind of vaccine space, but maybe for phase 2 vaccines, that I can't say never either.
Thank you. And likewise, so the adaptive platforms trials that you have presented, the paradigm for TB, present a major methodological leap. And how can regulators and national TB programs better integrate the findings from such trials into policy updates, for instance?
That's a really good question. I think in some ways we're all learning from each other, and I think in some ways, you know, what we're trying to do is we're trying to move towards innovation. And I think, you know, the rigidity of regulators is there for a reason and for a purpose. And Andreas was touching on that previously, and a previous comment he was making. So obviously, you know, first and foremost, it's safety. And I think, you know, you cannot forego safety, but at the same time, you know, we are trying to develop these drugs and regimens as quickly as possible. And obviously, you know, in some ways, we need to find innovative ways to not only develop these drugs, but also generate the evidence that the regulators, the WHO, and countries require in order for us to really get these drugs and regimens to patients. And I think, you know, here there are some barriers and obstacles, but I think thanks to Unite 40B, and thanks to Relay, and other initiatives, the WHO is very open. I have to say thank you very much, Fouad, for your input and your time and your team's input and passion in really helping asset owners, academics develop the drugs. So we have input from the WHO, which is really, really helpful, and we have met with the regulators as well. And I think it's through these interactions with the WHO, the regulators, while we are generating evidence and having multiple pit stops where we're talking to each other and we're communicating what we see and where we want to go, I think that does help quite a lot. So I think it's a journey at the moment. We all have the passion, we all have the desire to improve TB outcomes. I think that comes from the WHO, the regulators, but obviously from the drug asset owners. We all want the same thing. And I think there is where there's a will, there's a way. So I think, you know, we are communicating with each other. I think maybe that wasn't happening before. And so my hope is that, you know, through more communication, through these shared principles, we'll be able to accelerate and get through some of these regulatory barriers, improve our endpoints, find biomarkers which can generate better evidence, and not necessarily wait for clinical relapse in order to understand whether a regimen works or not. And I'll leave to Fouad. He may wish to add something along that. Thank you.
Well, maybe just to add quickly, thank you, Simon, that we definitely looking, especially now with this new financial environment in public health, for finding some efficiencies and trying to accelerate the very responsible and safe research to reach the stage where it can be already approved by the regulators and be reviewed for the policy and actual use by the national TB programs. There are some ways to do that. Of course, we are missing some pieces like the better biomarkers that make the like for the treatment trials make people wait and observe for quite a duration of time to see if there is a relapse. That's a major barrier, especially for shortening the treatment, because you cannot avoid that step, and that renders trials longer than they could be with some really good biomarker. But I should also say, and I showed in my slides, that the participation of countries and the sites in this clinical research, especially at the late stages, it's really important, because that can decrease the time from the research to actually countries adopting the innovation, be it the treatment or diagnostic, especially in the treatment side, when there is a potentially direct safety issues that countries, if they are not part of the late stage clinical research, sometimes tend to try and redo that research, which takes a kind of an additional loop of time, which we've seen especially recently. Now that we have a lot of things happening, especially on the TB treatment side, as I showed, and everyone on this panel knows that we have quite a big number of the new medicines potentially reaching the later stages of the clinical development. This kind of acceleration and finding efficiencies and not overdoing things, as Andreas said, for no obvious reasons, but still keeping this ethical and safe, would be important. And I think the community is of researchers, WHO, and manufacturers is really trying to make those improvements, which is very welcome.
Thank you very much, Fred. Maybe a last question that will help us as well, given the context that we are. Michelle, you have alerted that there might be further investment further commitment from the public side in order to further develop the molecule. So can you share with us what are the lessons that can be drawn from these public-private partnerships behind the development of Avibacters, and how this collaborative model could be expanded to other TB innovations, for instance, for all of you? That is for all of you, actually. If there are some lessons learned.
Well, one lesson is don't hesitate and reach out directly. Don't wait for the opportunity until maybe funding agencies come to you, but do it the other way around. I mean, it's not always easy, but you need to present your idea upfront in order to actually get some interest in people. And of course, when we started with TRIC-TB, we didn't know where it ends up. And if you have positive results, it's always easy to get a next funding vehicle because you somehow need to show sustainability. And I think this is the big asset of all these consortia, that we do not stop after phase 1 or after phase 2 because of the sustainability of all these fundings. We actually will be able, hopefully, to move this project really until the very end of the clinics. And that's, I think, hasn't been the case before, where you got partial funding for specific sequences of your development. And now it's much more interactive and much more, collaborative.
Thank you very much. And with that, this webinar will come to an end. And as Andreas and Simon has presented the next steps, then we give you a date for another webinar where you'll be able to present the results that we're very much looking forward to, and hopefully getting the phase 3. The good news that you're getting into phase 3 clinical trials for further development of this promising combination therapy. So with that, I would like to thank all of you for your excellent presentation, setting the scene. I think that was very important. And this webinar will be available on IHI website as well as our YouTube channel in a few minutes, hours. And I wish you a very nice day. And thank you again for your participation.